All right, it's 12 o'clock on Wednesday. That means it's time for the Iowa Learning Farms Conservation webinar. Matt Helmers with Iowa State University, and really excited to be able to be back hosting after missing some time, and specifically last week for the Drainage Research Forum in Brookings, South Dakota. And really excited to hear from our speaker today. Our speaker is Dr. Sabrina Ruis. She's a research soil scientist with the USDA Agricultural Research Service at the National Laboratory for Agriculture and the Environment. We may have some people on that still remember NLA when it was referred to as the Soil Tilth Lab. So the lab formerly known as the Soil Tilth Lab. Uh, Dr. Ruiz is going to talk to us today about managing cover crops to achieve high biomass and improve soil health indicators. As always, if you have questions, please type those into the chat box and we'll get those to Dr. Ruiz at the end of the talk. With that, the virtual floor is all yours, Sabrina.
All right. Thank you for the introduction. Um, so I've been at NLA since January 2023, and before that, I was at University of Nebraska-Lincoln. So part of what I'm going to bring here today is a number of case studies where we can look at some different strategies to improve our cover crop biomass and therefore improve our soil health indicators. So what is soil health and how do we go about measuring that? Well, the NRCS defines soil health as the continued capacity of a soil to function as a vital living ecosystem that sustains plants, animals, and humans. And there are a number of soil health principles that the NRCS has depicted in a nice graphic here. And they're looking at minimizing disturbance. So that might be the use of no-till, maximizing diversity, maximizing soil cover, and maximizing your living roots. So we have these different principles, and the idea is that they are to mimic nature. And if we think about our native landscapes here in Iowa, we had a lot of tall grass prairie. So that tall grass prairie encompasses a much larger span of the year than our corn and soybean systems do in terms of how active or the activity level of those crops. So how do we go about assessing the ability of a soil to function? Well, we use soil properties as indicators. And here's a list of just some of the soil properties that we might use as indicators of soil health or function. We have our microbial biomass, which is an indication of population size. You can kind of think of that as something analogous to quantifying the number of frogs around a pond or the number of earthworms in the soil. Potentially mineralizable carbon and nitrogen, that's looking at microbial activity. So how active they are they running a marathon or are they just binging Netflix? Particulate organic matter, carbon and nitrogen, you can think of that as food for the microbes. Total organic carbon and nitrogen, that's your inherent soil fertility and your nutrient cycling capacity, your wet aggregate stability. That's an indicator of soil structural quality and water erosion potential. And so larger aggregates are going to be less likely to be lost through erosion compared to smaller aggregates. Our soil bulk density, which is an indicator of compaction. Our soil nutrients, and this is NP and K is just a few, an indicator of fertility. Our soil pH, acidity and nutrient availability, and then water retention and infiltration. So we're looking at water capture. So when it rains, is that water getting into the soil? And then is it being stored and released during dry periods? So there are a number of reasons why we want to use cover crops and rationale for trying to achieve high biomass production, reducing water and wind erosion, increasing plant diversity, reducing nutrient losses, improving soil health indicators and soil carbon, reducing weeds, enhancing soil fertility, and managing soil water. So we've got a lot of perks associated with cover crops. But the question is, are they producing enough biomass to support these benefits, and what kind of management do we need to achieve that? How stable is that biomass from year to year? And some have even looked at using cover crops in non-prime agricultural lands to rejuvenate that soil and build soil health and soil carbon. For our talk today, I'm going to focus primarily on number 1 and 2, getting that biomass, and how do we manage the cover crops to achieve that biomass. So there are a number of factors that will influence cover crops and the soil health changes under those cover crops, including biomass production, duration of management, and that just means the number of years that you've used cover crop species, soil organic matter, soil texture, and others. I'm going to focus primarily on biomass production and duration of management. Cover crop species, much of what I'm going to present today is from cereal rye, and as it tends to be one of the more successful overwintering cover crops in the area. So when you start looking at the literature, and there are several papers that have done global or countrywide reviews of cover crop biomass and their effects on different aspects of soil function or their cover crop benefits. So that these are some threshold biomass levels that they proposed in these papers. About 0.6 tons per acre to reduce erosion, about 0.7 tons to reduce nitrate leaching by 50%, about 0.9 tons per acre to increase soil carbon, 1.7 tons to control weeds, and 2.7 tons to reduce nitrate leaching by 80%. So there are a lot of different factors that are driving the cover crop biomass production. And one of the things we want to know is, well, how do we get these different biomass levels? And some of it's going to depend on your goal for your cover crop. So we're going to look at 3 different general strategies of managing cover crops. First is planting early, and that might be pre-harvest interseeding or overseeding and then comparing that to our post-harvest drilled rye cover crop, and that will be in continuous corn and corn soybean. Strategy 2 is a late termination, which is right at corn planting or shortly thereafter, versus an early termination, which is 2 to 3 weeks before corn planting. And then strategy 3, which is looking at a cover crop following a short season crop. And those short season crops might be corn silage, small grain, high moisture corn. So our first strategy is looking at early versus late terminated cover crops. So we have a cash crop of continuous corn. We have 2 sites here. One is rain-fed and one is irrigated. At the irrigated site, the cover crops are not irrigated. And both of these sites are in the eastern part of Nebraska. We are looking at cereal rye. We've been doing cover crops for 6 years in this study. And as you look at these top 2 figures here, so when you're comparing the different bars within a year, you'll note here that this white bar in the 2014 is an A versus the black bar is a B. That means they are different. The biomass levels are different statistically. If both of them were A's, that would mean that they are not different statistically. So here at the rain-fed site, you'll notice that they're all A's for the late terminated cover crop and all B's for the early terminated cover crop. And even across the 6-year study, we have A versus B. So our late terminated cover crop increased biomass production compared to early terminated. If we look at the irrigated site, we see the same pattern. But you'll notice that the biomass level is much higher at this site. And just for conversion purposes, 1 megagram per hectare of biomass is equal to 0.44 tons per acre. So if we look at the number of years where we had biomass above that level, it'd be 3 years for the rain-fed site and 6 for the irrigated site. So now if we look at our soil health indicators after 6 years from the site, early terminated, so I'm only presenting here data from the late terminated site for the soil health indicators, because at the early terminated site or rain-fed site, there was no effect of the cover crops, whether it was late terminated or early terminated. So this is just for the irrigated site. Early terminated cover crop had no effect compared to no cover crop for all of these soil health indicators: our plant available water or water retention, water infiltration, soil aggregation, organic matter, and microbial biomass. But if we look at the late terminated cover crop, we do see increases in organic matter and soil aggregation. And now this data is for the 0 to 2 inch depth. And if we were to look deeper, and we did in this study, we actually didn't see any effects of cover crops below that 2 inch depth. You'll note that at the rain-fed site, we had this nice high biomass in 2015 to 2017, and then it dropped off. We did sample in 2016 at both sites, so that'd be 3 years of management. And we did see some changes in our labile organic matter and aggregation at this rain-fed site, but those went away by year 6 just because we did not keep the biomass high with time through that site. But at the irrigated site, because the biomass stayed, we're looking at above 2 for most years, that any changes that we saw in the organic matter aggregation, they stayed with that time. So it really highlights the importance of maintaining sufficient biomass from year to year as best as you can. I know we are subject to weather conditions. So strategy 2, overseeding or interseeding early versus your traditional drilling after rye harvest. And so we're going to work with 2 studies here. We've got one from Marcos that was published in 23, and then that took place here in Iowa. The one by Coiler Cole here in 2020, that took place in eastern Nebraska. We're looking at corn, soybean with cereal rice or cover crop. Now our cover crop seeding dates are mid-September for the early planted or overseeded cover crop, and mid to late October for the traditional drill seeding after harvest. So on the top part of this table here, we're looking at the study by Marcos et al. Here in Iowa. We have our broadcast and overseed and our drilled planting methods. And here's our rye cover crop biomass production: 0.07 to 2.79 tons per acre for the broadcast, and 0.03 to 0.44 for the drilled. So overall, we're looking at 4 or 5 site years, and a site year is the number of sites multiplied by the number of years. So if you have 2 sites and 3 years of data, that is 6 site years. So we have where the broadcast actually improved biomass over the drill treatment. If we look at the Nebraska study by Coiler Cole here at the bottom, at the eastern Nebraska site, we saw biomass anywhere from 0.75 to 0.99 tons per acre under broadcast, which was more than the drilled treatment at 0.33 to 0.44. However, when we go to eastern south central Nebraska, we're looking at higher biomass with the drilled compared to the broadcast. And so this paper discussed why there were those differences between those 2 sites, and it was attributed to rainfall patterns in the fall, with the eastern Nebraska site receiving more rainfall in the fall when we're trying to establish and broadcast these cover crops. And this This is just a picture of a high clearance cedar getting ready to go through corn and plant cover crops. They are adding, this is before they added the drop tubes on, but they are adding, you can see a couple of the drop tubes here, and they just haven't put the other ones on yet. And then this is a cover crop that was established right before, or planted right before a nice rain event. And this is right when we were getting ready to harvest. So we have a very nice stand here. So one of those studies that I had just mentioned with the overseeding, I was also involved in looking at some of the soil health metrics. And so this is the same Eastern Nebraska and Eastern South Central Nebraska study by Coyle-Cole. And so we measured soil particulate or labile organic matter, total organic matter, soil irrigation, water infiltration, and bulk density. And when we compare the broadcast cover crop to the drilled cover crop, we did see an increase in particulate or labile organic matter at one site and soil irrigation at one site. However, when we compare just cover crop versus no cover crop, we only saw an increase in soil aggregation. And our third strategy, planting a cover crop following a short season crop, whether that's corn silage, high moisture corn, small grain, or something else. So here we have a cereal rye cover crop stand in mid-spring near Boone, Iowa. And this is close to termination that year. This photo here is actually from a cereal rye cover crop that was planted after corn silage near North Platte, Nebraska. And I don't know, that's many hours away, but we do have potential to achieve this impressive biomass here from following corn silage. And it was just a nice way to convey how much biomass we can get with cereal rye. So here I'm going to show results from a study that was actually near Boone, Iowa. It was under corn grain or silage, followed by soybean. And then the cover crop was cereal rye again. The duration was 21 years. So that's 21 years of cover crops. And so if we look at the number of times that biomass was above that 1 ton or 1 megagram per hectare or 0.44 tons per acre, in all years following corn silage and corn grain, the biomass was above that threshold. About half of the years following soybean. So when we look at the cover crop biomass, when we compare the cover crop effect under grain corn compared to grain corn with a cover crop versus grain corn without a cover crop, we did see an increase in microbial biomass, but not soil organic matter or soil irrigation or carbon or nitrogen mineralization. And some of that is just due to the amount of corn residue that it has to compete with in order to establish that cover crop. And then when we compare a cover crop with following corn silage compared to corn silage without a cover crop, the corn silage with the cover crop increased all of these parameters compared to corn silage without the cover crop. And some of that is because we are, with corn silage, we are removing all of that residue that would otherwise be there over the winter. So we're leaving the soil bare and subjecting it to all of the temperature and moisture fluctuations that can wreak havoc on soil aggregation. Thinking of freeze-thaw cycles, if you've ever left a glass of or a jar of water outside in the wintertime and it's frozen and the glass burst. Similar principles happen with soil aggregates. And it's also hard on the microbial community. Just if you think about your house and you like to keep it at a relatively steady temperature, well, microbes would like a steady temperature as much as they could get as well to fulfill their biological processes. So I would be remiss if I did not talk about yields, because yields are important. They're why we're doing what we're doing. So here, from the studies that I presented, I have just the overall effect of the cover crop management strategy on yields. So here, termination timing in the Marcos study had no effect on corn yield relative to control. The broadcast or early planted reduced corn yield or crop yield in 3 of 5 site years. In the late terminated, early terminated sites in Nebraska, we saw no effect of late termination on corn plant, no negative effect, I should say, on corn grain yield in 4 of 6 years. With the corn silage study that I just discussed, no effect on any of the 3 crops across the 21 years. Now that's the mean across years. It is not looking at individual years. The broadcast or early planted in eastern Nebraska, 6 of 8 site years, had no effect for soybean yields, and all years for corn in the corn soybean rotation. And then similar in the south central Nebraska site, no effect in most or all years. So overall, we're not detecting a statistically different effect of the rye cover crop on cash crop yields. So why did these strategies work? Well, we saw that late planting was successful to achieve biomass and changes in soil health indicators. Same with planting after a short season crop. Planting early, well, because that is so dependent on precipitation, it can be a little hit and miss to get that biomass. And then, so not only is precipitation important, but the amount of growing degree days, or GDD, which is a measure of basically heat accumulation. So here we're looking at a study done by Crespo et al. And this is from the same study where I presented the 21 years of cover crop effects on soil health. And so she looked at the number of growing degree days with soybean followed by rye after both phases of the rotation, similar with silage, and then soybean with rye following soybean only, and silage with rye following silage only. And so she found that if we look at the fall growing degree days, so the time from time of planting to the cessation of growth, if the cover crop accumulated at least 800 growing degree days, that was a predictor of high biomass come spring. But spring growing degree days and total growing degree days were also important, and you can see the points are pretty tightly clustered around the dashed line here, which means it's a pretty good relationship. And related to that, so remember I was talking about the late termination, and that was planted in October. So that system's going to focus more on getting those growing degree days in the spring. And we do see that the more growing degree days you get, the better and this was total across the entire cover crop period, the more biomass you get. And then precipitation's also involved there too. If you don't have enough precipitation, the plants can't complete their photosynthesize. So we have to have a balance of both. So root biomass production is something that is really important because roots directly interact with the soil and can influence soil health. And so we have been, for the last 2 years, collecting 4-inch cores to 4-foot depth. And this is just a picture of one of those cores. You can't see the cover crop at the top here just because we cut it at the soil level before we take the core. And so here within this graph, you can see the 2023 biomass, root biomass with depth in red, and in blue for 2024. And so if we look at the total biomass production, we had just over a ton in 2023 and just over 3/4 of a ton in 2024. And you'll note that most of the biomass, root biomass, is in the top 2 to 4 inches. And so remember when I was talking about the late terminated cover crop and I said, yeah, we're just measuring the top 2 inches. And that's where we were only seeing, and we did measure the 2 to 4 inch depth and we were not seeing effects down that deep. Well, that's partly because we see most of the biomass at the top. So when we think about the native ecosystem, tallgrass prairie, this is a picture that I took at Neil Smith National Wildlife Refuge just few weeks ago, actually. And we've got a lot of biomass here. It's dense and a lot of it's perennial. So we have really extensive root systems here, but I'm going to talk a little bit more about this above ground and how it relates to the cover crop biomass production. So if we look at the native prairie above ground biomass, roughly 5 tons per acre. But when we look at our corn soybean, now this is the corn residue and soybean residue averaged across the rotation. So that averages out to about 4 tons per acre. So we're short biomass, above ground biomass input compared to what we had from a native prairie. Well, what if we add a corn soybean rotation with a 1/2 ton of biomass per acre per year from a cover crop? Well, then we're getting about 4.5 tons. We're closer to that 5.4 from the native prairie. And here, if we get a 1.5 tons per acre of biomass with our cover crop, the system as a whole annually is about 5.5 tons per acre, which is comparable to the native prairie. And so you're probably thinking, oh, Sabrina, you've been telling me about cover crop biomass and cover crop biomass. How do I measure it? So the USDA NRCS in 2018 put out this estimating cover crop biomass publication. So if you type in estimating cover crop biomass into Google or whatever search engine you want to use, this PDF will pop up and you can, it has different methods of quantifying your cover crop biomass in the field. Some general rules of thumb that are going to depend on the cover crop stand and tillering. 6 to 8 inch tall rye, about 800 pounds per acre. 12 to 15 inch rye, 0.75 to 1 ton per acre. 24 inch rye, about 2 tons per acre. And so just so you kind of understand where I pulled these numbers from for the biomass, the native prairie biomass was from Guzman and Alkaisi, that was published in 2010, and that was from the Neil Smith Wildlife Refuge. And then I used the county averages of corn and soybean and the harvest index from this paper by Ordonez to compute the amount of corn and soybean biomass, which was 5.3 tons per acre for corn and 2.6 for soybean. And then again, I averaged that to come up with this value. So in short, the closer we can get to our what we would have had as a native biomass input for biomass, the better. So take home messages. What do I want you to get out of this, out of what I've been talking about here? So to achieve high biomass, you need enough growing degree days and precipitation, and that precipitation is going to be critical if you are broadcast or interseeding your cover crops. And some ways to get that high biomass is by terminating late, or trying to plant early, or planting after a short season crop. If you want to achieve soil health benefits, you need that high biomass consistently. Remember the 6-year study from Nebraska? When we didn't have it regularly, we didn't see changes in soil health, or we lost those changes in soil health. Duration of management is important. It can take 5 years or more to see benefits. And that comes up multiple times in the literature of, yeah, it's usually 5, 6, 7 years before you really see benefits. Sometimes you might see them a little sooner depending on the soil and the degree of change that it is seeing. It can be more difficult to see changes in soil health in our highly fertile soils in this area. But that's not to say that cover crops have no purpose in these soils, because they do. Think about weed control, or the reduction in nitrate leaching, or the erosion control. So they are important even in highly fertile soils. What about effects on yields? Well, it depends on the weather conditions. If you get a dry period at a critical time in corn development or soybean development, yeah, you may see a reduction in yield. But overall, from that data I presented, it was generally that didn't affect yield. Don't overlook the importance of roots. They are in the soil, directly interacting with that soil. The more roots we can get, the better for soil health. And just remember, our overall goal is to mimic nature by including that living root year round, which is our cover crop. And we want to manage that cover crop to achieve high biomass and, of course, our crop yields. And be flexible. Our weather is all but routine here in Iowa. We've had dry years, we've had wet years in my time here in Iowa and my time in Nebraska. So with that, I will open it up for questions. And I think Matt's going to.
There we go. Okay.
Jump
I was
in here.
muted there for a second. Okay. I'm back now. Okay. If we can go to the last couple slides. All right. If you watch this webinar live and wanting to earn a CCA CEU, please email Elena Whitaker, ALENAW at iastate.edu by 5 o'clock today. Include your name, the name you entered to watch the webinar, and your CCA number. Again, the email to send to is ALENAW at iastate.edu. Let's go to the next slide. If you watch this webinar live and are willing to fill out a voluntary demographic survey, we'd greatly appreciate it. And you can scan that QR code or go to iowalearningfarms.org/survey. Only one response is needed for all 2025 webinars, excuse me. If you've done so, thank you very much. And if you're willing, we'd greatly appreciate it. Okay, next slide is probably, okay, we got Jake Wilson, that's an environmental scientist with Shive Hattery. From impact to improvement, how mitigation banking supports Iowa's aquatic resources. So a little different topic, but important nonetheless. And last slide should be that if you watch this webinar live and know somebody that would be interested, it will be archived on the iowalearningfarms.org/webinars page or on YouTube. So please go and watch that or forward it to somebody that you think might be of interest. Okay, we've got lots of questions coming in, but I'm going to take my prerogative as a moderator to ask two questions. Well, three questions, but I think a couple at least are quick. What method did you use for measuring infiltration?
So in the Coiler, or the study from that involved Coiler coal, that was the double ring infiltrometer.
Okay.
The late termination, early termination was just what we call the sorptivity test. So it's just that initial water infiltration.
Okay, cool. And then the study from Iowa with the 24 years, was that all no-till?
Yes, that was no-till.
Okay, cool. And then my last one is thinking about your kind of your budgets of biomass. Have you put that together where you include all the root biomass, looking at the root biomass of corn and beans and cover crop and prairie?
I have not, but that is a very interesting question. And a lot of people have struggled to measure roots just because it is laborious. Once you get
Yes.
that core and section it into pieces, you have to separate the roots from that.
Yeah.
And we do have a root washer, but it is
Yeah.
laborious.
Yeah. I hadn't seen it presented that way. I thought that was really useful to me. Okay. But I was thinking, oh yeah, well, but what about those roots below ground? So,
Mm-hmm.
yeah.
Yeah.
Okay. Sorry. All right. No more questions for me, but we have many questions coming in here. There's one good point. You talked about the silage, but seed corn fields might also be somewhat similar to those silage fields, a great potential to improve, to get biomass growth and improve some of those soil health indicators.
Mm-hmm.
Um, there was somebody that when you measured the biomass, did you also measure cover crop height to kind of relate to kind of how well those NRCS
No, we did
indicators?
not.
Okay. Yeah, I'll just have to say you're not the only one. I think about it now. I don't think we've done it either on our field. So, um, okay, what was the fertilizer source for the fields study?
Oh, so it depends on the study. In the rain-fed Nebraska site, it was anhydrous ammonia. For the one in eastern south central Nebraska, that was UAN. I believe the 21-year silage study was also UAN.
Yeah. Okay. Okay. There was one here on the why there were fewer years where biomass was greater than 1 milligram or megagram per hectare following soybeans, where there would be less residue to compete with for establishment or growing.
Yeah, that's an interesting question, and I think some of it has to do with the amount of soil moisture.
Uh,
Um, with corn, you're going to have a lot more residue to kind of protect that soil and maintain that soil moisture a little bit more than under soybean.
okay, great. Um, there was one here that at least some of the fields look like fairly flat fields. Can you project any benefits uh, for rye cover on more sloping fields?
That is a great question. And um, some of the people that I was working with at UNL um, submitted a grant to look at soil health and um, water erosion on sloping fields. To my knowledge, that grant is still in progress. Um, and they were looking at early planting and late termination. So they're trying to combine those two practices. Um, if I we did have a kind of an initial like pilot study, I would say it was funded by the Nebraska Corn Board, and we did have a sloping site there, and the the cover crops tended to do pretty well most years, and it, improvement in soil health indicators. It was short term, so we didn't see a lot of changes. But I would anticipate that with consistent use, we would see improvements in soil health indicators and reductions in erosion because we've improved soil health as well.
Yeah. I'm kind of reminded of, I think, a point that Dr. Rick Cruz had made before is that the first step in improving soil health is reducing soil erosion. So,
Yes.
yeah,
Yeah.
I don't know if it relates to this, but yeah. Um, all right. Next one was native prairie has far more diversity than the corn, soybean, cereal, rye cover crop. How might that plant diversity affect soil health?
So with diversity, so I'm going to take that from the standpoint of having like a diverse cover crop mix. And there it was a really nice meta-analysis or review of studies that compared cover crop mixes versus their single species counterparts. And in general, they found that there was no difference between having a mix versus a single
Okay.
species.
Okay. And there was one here about kind of cover crop rye tie up with nitrogen on especially some of these studies where you've not seen any yield impacts. Was there any kind of additional nitrogen management to try to overcome any potential nitrogen tie up?
We did not do any additional end management or any side dressing that was beyond what we would have normally done. Um, that would be definitely something to look into. I know there are some studies that have looked at the decomposition of the rye and kind of the end release as it decomposes. And some studies have shown that that end release can be in sync with crop needs, and others not so much.
Not so much. Okay. Here's an interesting one. Maybe I don't know. I'll ask it and we'll see if you have an answer or if we have to refer them somewhere else. In a year with fall armyworm issues, would we still expect early rye seeding to produce more biomass? They indicate there's more early growth to hopefully survive the feeding, but a much longer opportunity for feeding.
That's a good question. And I don't know that I have an actual answer because I haven't dealt with armyworm infestations in my cover crop fields before.
Yeah. Okay. Yeah, I hadn't heard that one. That's interesting. Okay, here's one kind of thinking about that shoot to root ratio of cereal rye. They wondered what is the shoot to root ratio of cereal rye at 1 megagram per hectare of above ground biomass? And does that ratio change as cereal rye grows?
Oh, that's a great question. Um, so I have seen cover crop biomass, let's say 1, 2 megagrams per hectare. And if you look at the roots in the, let's say, top 4 inches or so,
Hmm.
um, that can be, that might be about 1 megagram. It kind of depends
Okay.
on the seeding rate. Um, the the root biomass that I showed that had 1 or half a ton per acre of biomass, I believe that the above ground biomass was right around 1 or 1.5 megagrams per
Okay.
hectare.
Okay. But yeah, definitely that the root we shouldn't completely discount the biomass from that root. Yeah.
Yeah.
Um, have you done any research on fertilizer applications for the cover crop and looked at the impact on biomass production? If a goal was to harvest that cover crop or harvest that cover crop, I guess.
Mm-hmm. That is a great question. And we do have an experiment that is ongoing that is comparing a no-till fertilized rye against a no-till standard rye without the fertilization. And so some of that research is, like I said, ongoing, and stay tuned and we can
Okay.
add some more info to that question later as we go
Yeah.
along.
And somebody asked, but in what you presented today on cover crops, those were not fertilized.
Correct.
Okay. Yep. There was somebody along the lines of the multi-species cover. Is there any increase in microbial populations, not just total quantity, but species of microbes
Hmm.
with multi-species cover crops?
So I don't know. I would have to look into that one to see, just kind of how people are partitioning the species and the comparison between a single species versus multi-species.
Okay. I think you mentioned this, whether there's any work being done on timing of nutrient release from cover crops. And it sounded like you've seen some different results, and I don't know if you
Yeah.
all at NLA are doing any work on that now.
At the moment, not that I'm aware of, there is a plan to eventually look at and release for some legume cover crops that are
Okay.
following a small grain before
Okay.
corn. And so we're still working on establishing that experiment to really delve into that a
Okay.
little bit more.
Here was one thinking that we got some people interested in roots. So they asked which cover crop might have the most roots or biggest roots.
Well, I would just from observing cover crops in the field, I don't have data at my fingertips to really um, to look or to support, but a like a radish is going to have a larger diameter root. Um, And they can go fairly deep depending on the soil and how long they've been growing. But in terms of like overall abundance of roots,
Mhm.
that would be a lot of times a small grain like cereal rye or even oats if it gets enough time to grow in the fall.
Somebody, I'll just note it here. It's not as much of a question, but they indicated, please include monitoring for water quality in the ongoing experiment to see if additional fertilizer is impacting surface or groundwater. And I will say in our relay intercropping where we're growing rye and then planting soybeans into that standing rye and harvesting the rye and soybeans, where we're fertilizing that rye a bit, we're doing that. So we're measuring the water quality impacts of that.
And we are too. So
Great. Okay, cool. So we're, yeah. Okay. Um, oh, somebody asked if you could re-show that slide of the 0 to 2 and 2 to 4 inch depths showing the roots. They just were a little, maybe wanted a little more, see that again and information. And so it was, yeah, it was 0 to 2 and 0 to 4. So,
mhm.
somebody asked, is there a conversion factor of cover crop residue to organic matter?
Ooh. Um, I don't know that there would be a really direct one because it's so dependent on how that organic matter is stabilized in the soil and how the microbes are acting on it. Um, so in short, not that I'm aware of.
Okay. Okay. So on this one, maybe ask a question on this one with the 0 to 2 and 0 to 4. So for example, in 2023, 50% of the roots were in the 0 to 2 and 45 were in 0 to 4.
Mm-hmm.
So are there, I'm just thinking, would we expect more percent of the roots to be in the 0 to 4 than the 0 to 2?
Yes, and I think I might have those switched around. My apologies.
Okay, okay. Yep, okay. Just perhaps there. Yeah, but
Yeah,
so if I think about this, that most of them are in the 0 to 2, but there are some in that kind of 2 to 4 range. So
yeah, yeah.
okay, okay, cool. Thanks for that question. And now, yeah, I understand. I think I understand that better too. So okay. Um, Sabrina, I think that got to pretty much all of the questions that are in there. Uh, Thank you so very much for this important presentation, and you brought up some new studies that you have going on. So hopefully that means we can twist your arm again to come back and fill us in as part of the Iowa Learning Farm Conservation webinar series. Um, Folks, if you need a CCA CEU, please email Elena Whitaker, ALENAW, at iastate.edu by 5 o'clock today, including your name, the name you entered to watch the webinar, and your CCA number. Please tune in next week to hear about some of the mitigation work that's happening. And with that, I really appreciate everybody attending today. And Sabrina, thank you so very much for the work you're doing and for being willing to share with us today. So,
Yeah, no problem.
all right, everybody have a great day and rest of the week. Take care.